Recording. Oh, you want me to start recording now? I think I'm okay. So I don't know if I'm going to get back to my picture. Okay, so Google Meet. So I'm not talking to you face to face now, but you can hear me. What you want to do is, let's see if I can go to Top Hat. So let's see. Okay, can you see the Top Hat screen? I may have kicked you out. Okay. Okay. Could you see the Top Hat screen at all?
What?
Not
No.
now. Did you ever see it?
Yes.
Okay. So let me try again then. So we'll look at the Top Hat screen, and over to the side, there's this proctoral practice exam, and you need to click on that, and then it's going to ask you some questions. So can you answer this question? I suppose it's not scheduled yet. I don't know. This is not what I was expecting. Okay, I'm going to send you an email about this. It will give you the information you need to take the exam, what kind of browser you need, and a laptop or regular desktop computer. You can't do it on your iPhone or iPad, that kind of thing. So I'll send you an email about that. And then I'll ask more about this proctoral exam and why. I've got one question and that's it. Contains one item and worth 1 point. Okay, that's not going to help you at all. Okay, so I'll get that straightened out. For right now, let's go to, I did get the 2 lectures posted for Monday and Wednesday, but there was no Tuesday. We couldn't find it anywhere, so it was lost. I apologize for that. So we're going to go back to Sherwood, chapter 23. I'm sorry, chapter 3, and resume with slide number 6. Or I guess it was chapter 3. Yep. And we were on slide 6. Just before I get to that, when we go back and maybe have some coming up. Okay, so I'm just going to go ahead a little bit. when we see something like this is an MRI, but with color enhancement for the computer. This is a normal over here at A. This is a normal MRI. And then we have what's called functional magnetic resonance imaging. So those are gray. What we saw in chapter 3 for Martini, we saw a lot of these LMs, so light micrographs. And this is where you cut the tissue and you shine light through them. There's another type of image which is really highly magnified, and that is an electron micrograph, or EM for short. It's going to be gray, a scale like the MRI, but it's going to show you a higher magnification of the structures that you see in the light micrograph. So if we were going to look at a cell that had cilia on it, we could actually see the structure of the cilia and how it's moving and attached to the cell. We could see the detail in the nucleus. So it's a higher resolution. So maybe 40,000 times instead of 100 times, or 12,000 times, or 1,000 times. So it's a higher resolution. And I'm just saying that because it's covered in your earlier chapters 1 and 2. And I want you to be aware of that. Okay, so we're going to go back to Sherwood chapter 3 and resume talking about the adhesion proteins. And if you were looking at an electron micrograph, you could see the details of the desmosome, not the individual proteins, maybe really, really high magnification. You could see the cadhedrons, but usually what you'd have to do is label the protein and then you could visualize it. A desmosome is a cell attachment that has these proteins called adhesion molecules that are outside the plasma membrane. So plasma membrane is here. They go into the plasma membrane and then they attach to an intracellular protein and some cytoskeletal proteins. And the extracellular part interacts with the neighboring cells. So we have cell 1, we have cell 2, and these cadhedron molecules attach to each other in the intercellular space. This actually was the design that went to forming Velcro. So somebody was studying the desmosomal attachments and thought, hey, I can figure something out in my laboratory where I can have a fuzzy surface on one side and something with little hooks on it on the other side. When I put these two things together, I get the Velcro. So this person was actually studying cells, but came up with Velcro, and now he's probably a billionaire because everybody has Velcro. What's important about the desmosome is it attaches the cells very strongly to each other because of these keratin, usually keratin filaments. So if you want something really strong, then you use keratin, which is intermediate filament. If it's more delicate and you don't need reinforcement, then you can use the microfilament actin. But in the case of cells in your epidermis, you want that keratin because it's tough, and you don't want the the skin cells to come off with light frictional forces. You go into your mouth and you can get your fingernail and just lightly scrape the inside of your mouth and get cells pulled off. So the adhesion of the desmosomal attachment adhesion, I'm not positive, but I would guess probably you're not using the keratin, but you're using something that is more delicate, and that's why the tissue comes apart more easily. So keratin or actin and extracellular, so outside. So keratin is inside and actin would be inside. And outside, you then have something that's flexible but not elastic because you don't want the cells to get pulled apart. So you could use collagen. If you did want elasticity, then you could use elastin. And then when you're attaching cells to the basal lamina, you want them to stay put, then you use this protein called fibronectin, which actually translates to mean stay put. So desmosomal attachments, something that comes in the apical surface of the cell. So if we drew the top of the cell over here, if something came in through the apical surface, got into the cell, and you wanted to get it out, you could immediately transport it out into this intercellular space, and it could diffuse through the desmosome and get down to bloodstream. So it's not going to be a barrier for molecules that want to diffuse and get into the bloodstream. What it's there for is holding the cells together. But the next cell adhesion tells you tight junction. This type of attachment prevents the passage between cells. So this forces the substance. So let's get a color for our substance here. And, and we put it out here. We don't want it going between cells. So if it tries to go in between cells, it has to come right back out because a tight junction prevents it from going between the cells, the intercellular pathway. So now if you want to get that in, you have to, if you're going to allow it in, you have to have a transporter in the microvilli that will attach it and then bring it in. Once it's in, it can diffuse below the tight junction. There will be desmosomal attachments down below, but it can diffuse out right below the tight junction and then diffuse out the lateral side and go past the basal side and get into the bloodstream. And that's what you're showing. They're showing over here on the left is you've got selective uptake of substances through usually the surface of the microvilli will have the transporters, You don't have to get all the way down to the base of the microvilli as the arrow is implying. You can get it in right away. That's what the microvilli is there for, is to increase surface area for absorption. And not everything that you have in your lumen does your body want to have in its bloodstream. So there will be lack of transporters for things that you don't want, that could be in your food. Maybe the food is not digested enough, it's too big. You have to mix it more with your digestive enzymes. But there might be something that's totally indigestible, and that's your fiber, and it's not going to go in at all. It won't go between cells, and it won't be absorbed through the microvilli. So tight junctions are supposed to be tight, but sometimes they get messed up. And we'll talk about that in 302. So tight is, I guess you would say, it's relative. Thank you. So put some little quote marks there. They're tight, but they can become loose, and then you do get things going through the intercellular pathway. So what does a tight junction, what's it made up of? It's made up of claudin proteins, and they are occluding. So occluding and claudin proteins, they touch here. It said the kiss site, isn't that cute? And you have multiple kiss sites, multiple strands of occludin proteins. So if your tight junction number 1 breaks down, you've got 2, 3, and 4 that will prevent things getting in. If the second one breaks down, you got 3 and 4. If 3 and 4 break down, then something's going to go right past this tight junction and go between cells. And as I said, we'll talk in 302 about that particular problem, but you don't need to know about it at this time. You just need to know tight junctions. So tight junctions force the cells to be selectively permeable. The desmosomal attachment, if that was all you had, there's no selectivity. This stuff can go right through the lateral membrane passageway and into your body. So tight junctions force selective uptake, and these next junctions, they're called gap junctions. So gap junctions, they're designed so that the particle size, if it's less than 1.5 nanometers, like water and electrolytes, and hormones, they can go from one cell to the other using this gap junction. If it's electrical charge carried on an electrolyte, then it can pass from one cell to the next. So smooth muscle cells connected by gap junctions, they will contract pretty much at the same time, so you'll get that syncytium. You also will have your heart using these gap junctions so that the heart muscle contracts as a syncytium to create a pump action to move your blood. So down here, heart, smooth muscle, but also your primary and secondary spermatocytes and spermatids in the male, they're going to use tight junctions to share hormones and nutrients so that they will develop at the same rate. So from one stem cell, you can get 4 spermatocytes and spermatids formed. And this is very advantageous. You don't have to have a one-to-one stem cell to um, resulting sex cell production. You get 1 stem cell to 4 of the spermatids released into the seminiferous tubule and can exit. More about that in 302. Okay, now we're going to just talk about permeability. So if something is permeable, that means it can cross the plasma membrane. But how is it crossing? That can be in various ways. Sometimes it diffuses across the plasma membrane. We go back to the fluid mosaic model here. If something is lipid soluble, it can go right through the plasma membrane because of the phospholipids and cholesterol. So it has to still be slightly water soluble to get to the plasma membrane. But once it gets to the lipid part of the membrane, it goes right on through. So an example of something that can come in that's lipid soluble is alcohol. So if we were running a lab, we would, in 302, we will run this lab. We would have red blood cells and we put them in 3 types of alcohol: ethanol, methanol, and propyl alcohol. And we'd see how long the cell survives having alcohol in solution coming into the cell. Because the alcohol being permeable through the plasma membrane is going to build up inside and decrease water concentration inside, which is going to pull water in through channels and in through the plasma membrane and rupture the cell. So permeable could be diffusing through, and it could be carried across, and it could be active transport. Something, so we're going to be talking about simple diffusion, and we're going to be talking about what's called, secondary active transport, which is really diffusion, but it's depending upon a gradient that's established by active transport. So secondary active transport is really diffusion. It's usually carrier mediated, so you have to have a protein that's going to move this substance. But active transport, when we talk about primary active transport, you have a protein, but it uses ATP. It uses energy. This primary active transport is oftentimes what establishes the gradient, concentration gradient for secondary active transport. We have some things that are considered impermeable, even though they do get into the cell, because they're either, number one, consumed when they go in. An example of that is glucose. We know that glucose goes into cells. We know that glucose goes into red blood cells. But if we put red blood cells into glucose concentration that's equal to the solute concentration of the red blood cell, nothing happens. If we put a red blood cell into sodium chloride, they both come apart in solution. Sodium and chloride, after they break apart, they're both permeable to the plasma membrane, but they have a primary active transport that's going to pump them back out. So we've got chloride active transport that'll get the chloride pumped back out. We've got a sodium active transport that will pump the sodium back out. So since the sodium and the chloride don't build up in the cell, but are pumped back out, they're considered to be impermeable. We know they're not. They go in. We have the channels. We can prove it, but they don't stay inside the cell. If the active transport is working, they're pumped back out. You poison that ATPase, the enzyme that uses ATP to pump the sodium back out, then yeah, it will build up inside the cell. So, why is that up there anyway? Okay. Membranes are selectively permeable. You have to either be lipid soluble to get in, or you go through a channel to get in, or come back to this picture. You're lipid soluble, like alcohols, you can go in. You have sodium channel, you can come in. You have a transporter over here, and that brings you in, like glucose. Glucose has its transporter. And so glucose out here, let's change to that. Glucose comes in, binds, and then gets transported in, and we've got glucose in the cell. Now, a red blood cell is going to use glucose because it doesn't have mitochondria. So it's going to send the glucose into glycolysis. And when the glucose is broken down in glycolysis, it's not building, it's being consumed and made into ATP for the cell's function. So it's not building up inside the cell. It can't change the concentration gradient of solute inside the cell and doesn't have any effect on the gradient for water to move in and out of the cell. So glucose is said to be impermeable. Sodium chloride is said to be impermeable. The big things, big proteins can't get in. Enzymes usually can't get in. They have to attach to something. Hormones have to attach to something on the outside to activate the cell. If something goes into the cell, it changes the concentration gradient of solutes inside the cell, unless it's pumped out or consumed. If something goes in and builds up inside the cell, it's going to change the concentration gradient inside the cell for solute, and then water is going to move. Water is going to move first before other solutes can move to try and equilibrate the concentration gradient. Now, most cells can try and save their lives so they don't explode by moving solute out, so preventing water moving in. But if something is so permeable, like an alcohol, it goes in really quickly because it's in a high concentration, then water is going to follow and the cell is going to lose its battle and it's going to explode. So permeable via phospholipid, it has to be lipid-like. It has to be lipophilic. So if it's going through a protein channel, it has to be hydrophilic. Because the amino acids on the protein will have charges. If they're a channel, then water is going to be in that channel as well. So they have to be hydrophilic. The unassisted transport and then assisted transport. If a material can pass through a channel, like a sodium channel, then it can do so with no help. But if it's too big, like glucose, too big to get in through a channel, then it has to have some assistance. It has to be helped, which the term is called facilitated. Facilitation means to help. So being helped means you have a transport mechanism. Transport mechanisms, as I mentioned earlier, if they're passive, then no energy, no ATP is used by that protein itself. But if it's active transport, then ATP is required. So we're going to talk about active transport. So we have primary active transport. And if it's passive transport where no ATP is used, then this is secondary active transport. So think of using the words secondary to. That means as a result of something that happened. So while this protein is not directly using ATP, it's doing a passive movement. It's relying. So no energy is used, but the protein is relying, on a concentration gradient. Whoops. Why isn't it writing? Switch back to that. Okay. There. Well, now I have to erase it again because I'm not writing legibly. Okay, so concentration, sign for concentration is brackets. And then if we put GLU in the bracket, then we're talking about a glucose concentration gradient. If we put sodium in the bracket, we're talking about a sodium concentration gradient. Glucose might have its own transporter called a GLUT. So GLUT for glucose, and then T for transport. So a GLUT1 or a GLUT2 transporter. We also have sodium. It can go through its channel, and it's usually higher on the outside and higher on the inside. I'm sorry, lower on the inside. So sometimes we'll get glucose and sodium transported together so that the gradient for sodium is the tendency for sodium to come in is used to transport glucose in. So then we have what's called an SGLT transporter. So sodium, glucose, so GL, and then transporter T. And we've got various numbers of those too. These are either using the sodium gradient, which is maintained by a sodium and potassium ATPase, so that would be primary transport of sodium out to keep the sodium concentration low inside the cell. If we're using glucose transporters like a GLUT1 or a GLUT2, the glucose being consumed and turned into ATP is what's keeping the gradient low for the glucose transporters. Okay, so we either have passive, unassisted diffusion through a channel or lipid-soluble substance going right through the plasma membrane. Or we've got assisted and facilitated transport, which means we've got a protein that's helping the molecule get across. That assisted or facilitated transport is technically this passive transport. No energy is used, so we can, but if it's using a gradient that is established by primary active transport like sodium, then, and not glucose. Glucose isn't secondary active transport, but using ATP to maintain the sodium concentration low inside, then this would be secondary active transport. Okay, so let's look at a cell. So draw a cell. And what is the solute concentration inside a normal cell? It's about 300 milliosmoles. It's actually 290 milliosmoles, but they usually round it up to 300. In order for water not to move into the cell or out of the cell, so this is the ICF. The ECF must be at 290 or 300 milliosmoles. If the ECF changes, if the ECF concentration goes down, so if it goes down to 100 milliosmoles, then that means that you've got an increase in water concentration outside. And that means you have a gradient for water to enter the cell. Water can actually go through the lipid. If we go back to that cell membrane, since water is a dipole, it has a negative charge on the oxygen and positive charges on the hydrogen. It tumbles around. It's interacting with other water molecules and other solutes. And so it's constantly moving from one place to another. When it gets close enough to the phosphate heads, the hydrogen will attract to the phosphate head, and as it's spinning, the molecule will get in the spaces between the phosphate heads, and now its momentum will push it across, and then the hydrogen will be again attracted to the phosphate head on the inside, and it eventually gets into the cell. So how fast that's going to happen depends on how great a concentration gradient there is. And also we'll learn about some channels that are specific to water moving in, and these are aquaporins. So if you got a lot of aquaporins, water moves in really fast. If it's limited to going through the plasma membrane, it's going to happen slower. And actually, the way aquaporins were discovered is somebody was doing this simple experiment with red blood cells and putting them into different solutions and blowing them up. And when you put red blood cell into propyl alcohol, it will blow up in 3 to 5 seconds. And this researcher was thinking, well, that's really fast. If the water is just tumbling through the plasma membrane, how does it happen that fast? There must be something else water's going through besides the plasma membrane. So he went and searched the plasma membrane, and he found these water channels, and he called them aquaporins. The ability to get through a channel depends on the size of the molecule. If the molecule is charged, then the bigger it is, the slower it goes across. If it's like lipid, that means it's mostly nonpolar. That means it's got carbon, carbon, carbon chains on it. In the lipid soluble, it's got charges on it, positive and negative charges on it. And so it's got multiple charges. It's going, the larger this molecule is, the slower it's going to move across. The lipid soluble non-polar molecule, the more carbons it has, the more like the lipid tails of the phospholipid it is, and it actually moves across faster. So it's kind of counterintuitive. You think big people move slower than smaller, leaner people. Well, that's in the case of molecules, that only is true if it's water soluble. If it's hydrophilic, The larger the molecule is, the slower it moves. But if it's lipophilic, the larger the molecule is, the faster it goes into the lipid of the plasma membrane, and the faster it moves across the plasma membrane. The highly lipid soluble particles, they're actually dissolving in the lipid bilayer and moving across. So put here nonpolar molecules. The non-lipid particles, they require either assisted, so facilitated, transport, or if they're smaller, they can actually go through a membrane channel. So something like sodium and chloride can go through a membrane channel. If they're non-lipid, they're charged, that means they're polar. So ions like sodium, the ion, chloride ion. You can have potassium ion. So those are the ones that we're mostly going to be talking about. Glucose, also polar. It has its own facilitated transporter, or it can go with sodium through the SGLT transporters and get into the cell. So unassisted, that means it's passive, and it means there must be a concentration gradient involved. We use the term diffusion is going down a concentration gradient. So we've got our cell. If we have 100 milliosmoles out here and 300 milliosmoles inside the cell, we have high water concentration out here, low solute concentration. So this 100 milliosmoles is referring to the solute concentration. If there's low particles, then there's high water. If you have 300 milliosmoles in your cell, then you have high solute. It's normal, but it's high with respect to what is outside. So you have, with respect to what is inside the cell and solute being high, then you have low water concentration. So water moves first. That's what we said earlier. Water always moves first. Why? Because it's the smallest molecule around. So water is going to move down its concentration gradient. So down means from high to low. So water is going to enter the cell, and the cell membrane is going to expand unless the cell can move some of its solute out. So if you give the cell enough time, it takes some of that 300 and starts moving some particles out. So it reduces the 300, trying to get close to 100. But if the gradient is high enough for water, water moves in so fast before the cell can move solute out that the membrane continues to expand, and then pretty soon the membrane ruptures and you've lost your cell. Okay, so moving down a concentration gradient. Also, you can move down an electrical gradient. So inside of a cell has a net negative charge on it because of all the cytoskeletal proteins inside. The outside, I'm going to take away the dashes here. So where the concentration gradient could lead to rupture of the cell. Now we're just going to be talking about the electrical gradient. So the electrical gradient is the cell inside is negatively charged because of the proteins inside. It also is the negative charge is kept there because potassium, which is high inside, let me change the color here. Now let's go back here, get a green. We've got potassium high inside the cell and potassium low outside the cell. So we make a big K for high concentration and small k for low concentration. We have channels for potassium to leak out. And it would leak out down its concentration gradient from inside to outside. With potassium, it's got a concentration gradient to leave. So concentration-wise, it wants to leave, but electrically, since potassium is a positive charge and the inside of the cell is negatively charged, it wants to stay. So how much potassium gets out is determined by how permeable it is, and then also what the electrical charge is. So if we're talking about the permeability of potassium, potassium is fighting its concentration gradient with its electrical gradient. They don't work together, they work against each other. Now, if we look at sodium, and then we'll take a break. Sodium is, you got a big sodium outside and a small sodium inside. So that's the concentration gradient. Oops. So go back to the purple and add my 0 there again. Okay, so we're talking about a concentration now. So this side of the picture, we're just going to erase. It will be on the recording, but we're going to take it away right now. We're going to draw our plasma membrane again for our cell. We're going to be equal concentrations inside and outside. So 300 milliosmoles outside, or 290, whatever, and 300 inside. We still have a solute concentration that tends to have sodium coming in and potassium going out. Now, sodium's gradient, it's got a concentration gradient to enter, and it's attracted to the negative charges of the proteins. So it's got an electrical gradient to enter. So sodium has two things working in its favor to get into the cell. Potassium has a tendency to want to leave due to its concentration gradient, but stay in due to its electrical gradient. Now, there's one more factor that we have to consider, and that's permeability, overall permeability of the ions, and we'll introduce that later. But right now, we've got potassium tending to kind of stay in because of the electrical gradient and leave because of its concentration gradient. And we've got sodium that wants to enter down its concentration gradient and also down its electrical gradient. So two things are adding together. The sodium doesn't stay in. We talked about it being, go back to this slide number 10, it's acting as though it's non-permeable. So sodium is acting non-permeable because it's pumped back out. And potassium is pumped back in. So we've got off to the side, we've got ATPase. That's going to be taking potassium and pumping it back in. And it's going to take sodium and pump it out. The ATPase is pumping three sodiums out for every 2 potassiums it pumps back in. This inequality helps to keep the inside of the cell negative because it's not pumping back in as much potassium as is actually leaking out. Okay, so we'll take a break. Went a little bit long here. I'll come back at 11. So 11 minutes after 11, and we'll resume the lecture with slide 15. Okay, we're back. So, simple diffusion. Since it's a passive process and it's molecules that are randomly moving around in a solution, things can happen to that solution that will alter the activity of the molecules. So they're running into each other, they're running into maybe a container, they're bouncing around, and they're doing it at a certain speed. If you heat the protein up, if you heat the solution up, then the molecules bounce around faster. So it speeds up. If you cool it down, then that slows the motion of the molecules down. This random movement of the molecules is called Brownian motion. You might have run across that in chemistry, and also physics talks about Brownian motion. So the tendency for the molecules to bounce around, if they're in a gaseous state, then they spread out from an area where they're introduced, and you put your finger on your hairspray can and you see the particles diffuse out as the spray gets further away from the can. If you were to heat up that can, it has a warning on it: do not get close to heat. Why? Because the molecules speed up their motion and they put pressure on the inside of the can, and the can explode. So speeding up the molecules in an enclosed space can lead to problems. Um, Something getting colder contracts and slows down the motion. Normal body temperature, 37 degrees. Above 39, things start to speed up. Below 36, things start to get too cold and molecules don't move fast enough to get across plasma membranes. Another thing that is a problem with heat, since we have so many proteins in our body, if we heat up above 39 degrees, we can get proteins denaturing. So in your earlier chapters that you're supposed to read on your own, we know that there are amino acids in a chain, and they can then go into an alpha helix or into a beta cleavage sheet. And these can then get folded, and we have a tertiary structure for a protein. So primary structure, secondary structure. You fold the protein, and that leads to the tertiary structure. And then if we were to put several proteins together, we could have a quaternary structure like hemoglobin. So we've got this intricate folding going on, and we want that folding to be in place for the proper function of the protein. You heat it up, it starts to unfold, and that's the denature that's occurring, denaturation. So the proteins don't function properly. If you can cool down the protein before it totally unfolds, it can refold. But if it totally denatures, you've lost the function of that protein. So heat is one problem, also pH. So too acid or too basic, that can denature proteins as well. So remember, simple diffusion goes from high to low concentration. And also the concentration, we're talking about the solute concentration. But we can also, when we talk about the cell and we talk about milliosmoles, we're talking about solute, 300 solute concentration. Also, we have a water concentration to take into consideration. And since water moves first, if we have 300 here and we have 700 out here, that means relative to what's outside, we have low water and we have high water, and water is going to leave down its concentration gradient. So always have to think about what the story is, what the osmolarity is, and what does that mean as a consequence for water concentration. Water is going to move first. Water leaving a cell, the cell will shrink. If water was entering, the cell would swell and it might end up bursting. Okay, so net diffusion will stop if you reach equilibrium. Well, you can do that experiment in a lab. In real life, when you reach equilibrium of ions or equilibrium of proteins or whatever, then you've lost the ability to do work in your body. So we'll be talking about when net diffusion stops, but this will be in vitro. This won't be in vivo. That won't be in the body, but we'll use it as an example for teaching purposes. Diffusion down concentration gradients is crucial for the survival of every cell, so it's gonna happen. Diffusion of water can also be around cells. That's where this picture comes in. All these lines here are axons. They're lipid membranes of neurons. They're oriented in different, so there's a coronal plane, there's a horizontal plane, there's oblique planes. The axons are going to different destinations or coming from different locations. And what they're doing is looking at the diffusion of water around those axonal planes. So they've taken an MRI machine and now they've programmed it so that it will be able to look at the diffusion of water. And it also the program has assigned colors to the particular orientation of the white fiber tracks. So horizontal is greenish yellow, and vertical is the blue purple. You get into the oblique angles, kind of slightly off to the side. Then you've got the magenta. And what they're doing is, first of all, they know where the normal pathways are. And then they look at this diffusion tension imaging or DTI, and they're looking for abnormalities in the, the planes and how the water is moving. So they got pathways coming in from the spinal cord. They've got pathways coming out through the spinal cord and then going out to various locations. So frontal lobe, parietal lobe, occipital lobe. Down here, we've got the pons. Cerebellum seems to be cut off back here. So this MRI shows you some of the horizontal white fiber tracks. So they actually look white in this picture. And here they've colored the tracks so that the horizontal here is really nice yellow. And it's going from the back of the thalamus out and around and back to the occipital lobe for your visual cortex. And it's got some other pathways that are being depicted by different colors here. So this is one way to look at your brain. So your introductory course, you should know by now the corpus callosum, but we'll learn again at the neuronal part of our 301. This area here that is red going across this direction here, this is pons. This is anterior commissure. Don't have to know these. I'm just pointing some of the things out that you might remember from your introductory course. So corpus callosum is the largest of these commissures. We've got a commissure in the pons, and then this corpus callosum, meaning it's huge, colossal, it's huge. When we look at the blue, let's use that color, it might show up a little bit better. So we look at the fiber pathways, they're coming into this area and then projecting up and out. And then we've got some other pathways that are coming down and out using the same similar pathways. One that's going out is coming from the frontal lobe, primary motor cortex. The one that's coming in is going to the parietal, the primary sensory cortex. So frontal lobe, motor, that's why it's going out, and coming in, sensory. So again, they're looking for abnormalities in the pathway, and then they can do some diagnosis. So that's diffusion of water. Let's look at this introduction to diffusion, where we've got area A that has a high concentration of these orange balls. And we've got area B that has a low concentration of the orange balls. What is wrong with the discussion down here says diffusion is happening from area A to area B. What is wrong with this picture is water. They didn't talk about water. They're talking about orange particles. Oops. Diffusion is from A to B for the orange particles, but water, and water moves first. It's from B to A. So when I give you a problem, always think water first. Water's low in concentration here because there's particles in the way. So water is only very limited in its concentration because all these orange particles in the way. Water is much higher concentration here because there's fewer orange particles. So water moves down its concentration from B to A. And then later, second is diffusion of orange to from A to B. The diffusion will continue until you have equal distribution of water and orange particles. And then that will be your equilibrium and no net diffusion. So the term net means you've got particles moving and taking up space from A and moving into space in B. So you got this guy coming over here. Oops, he didn't change to orange. So you got him moving over. That's net diffusion, and that will continue until you got the equilibrium established. No net diffusion, but still, if one orange goes over, then another orange comes back. So there's still diffusion, there's still random movement of particles, but no net diffusion. So this guy, Fick, was looking at diffusion, and he came up with some information about diffusion, and it hasn't been disproved yet, so it's referred to as the law of diffusion. And his name is attached to it. It's Fick's law of diffusion. And he's summarizing what we've discussed so far, that there will always be a concentration gradient. So there will be an area of high solute concentration or low water concentration, and then in the other area, low solute, high water. The higher the concentration gradient is, the faster the rate of diffusion. So you increase the concentration gradient. You go, let's go back here. If I went up to 1200 milliosmoles, then water would leave the cell faster. And if this was a lipid-soluble substance, water is leaving, but this enters so fast that it starts to reverse the gradient for water, and water comes in, the cell could blow up. So the higher the concentration, the faster things diffuse. So that was his first edict. And then the larger the surface area for diffusion to occur, then the more times a particle can hit the membrane and greater chance for it to go across. So you increase surface area, you increase the rate of diffusion. If it's lipid soluble, then it goes through the plasma membranes phospholipid area and goes in really fast. So the more lipid soluble it is and the more like lipid is, the more carbons, then the greater the diffusion rate. The water soluble, so hydrophilic, here they're talking about the molecular weight of a hydrophilic molecule. The more it weighs, so you increase the size of that hydrophilic molecule, then diffusion rate goes down. If you make the membrane thicker, and sometimes in pathological situations, disease processes, you get fibrous connective tissue building up, then there's a thicker membrane to go through, or there's a thickness between where the blood vessels are and the cells are, then the diffusion rate goes down. So he came up with this equation here for net rate of diffusion. Now he's talking here about nonpolar, he takes the square root of the molecular weight. That's of a nonpolar molecule. He's got lipid solubility in the numerator. He's got diffusion distance. He's got molecular weight. These are going to decrease diffusion. He's got concentration gradient and surface area and lipid solubility in the numerator, and these are going to increase diffusion. So net diffusion, the greater the concentration gradient, the more surface area, the more lipid soluble it is, the faster the diffusion rate will be. The bigger the size of a nonpolar molecule, the slower it will be. And the thicker the membrane, the slower it will be. So concentrate on this and what each of these components are referring to in this equation that Fick came up with. Don't have to worry about the rest of this down here, so no worry. And then worry about this one. Yeah. So and what everything means up here. Okay, so membranes. We've got a membrane here and it's of constant thickness. And we've got pores in the membrane of a constant size. And we're saying that the green particles can actually go across. So we've got the green particles moving through the pores, so they can diffuse. So number one, we've got high solute, so therefore we have low water concentration. We've got low solute, so we've got high water concentration. So this arrow rung, first thing that happens is water moves from side B to side A. And then second, we have the solute, the particles going from A to B until we get to equilibrium, and then net diffusion stops. Here we've got side A and side B, but the pore size is too small for the molecule. So the molecules can't move, but again, we've got high solute. So we've got low water. We've got low solute concentration. So high water concentration. So what moves? Water moves from side B to side A. So water diffusion, but no solute diffusion. So water is going to move across until, and actually this membrane should probably extend up, because what's going to happen is the volume of side A is going to go up and the volume in side B is going to go down until the particles and water on each side of the membrane are at equal concentration. So water is going to change the solute concentration in A by moving over there and spreading the particles out. So ion channels, potassium, we saw that moves out of the cell. We saw sodium moves into the cell. Chloride also moves into the cell. The movement is just leaky. It's dependent on the permeability of the ion, and it's also dependent on the concentration and electrical gradients for those ions. A leak channel is always open, but some other channels have to be activated. So an activated channel is usually closed until it's, so at rest, it's closed. And when it's activated, this is very messy. When it's activated, then it will be opened. These, I mentioned yesterday, they can be activated by a ligand, such as a hormone or a transmitter, neurotransmitter. Or they can be electrically activated. So we'll be talking about electrical activation coming up shortly. We introduced electrical concentrations. We introduced the electrical gradient. Sodium moves down its electrical gradient from the positive charges on the outside of the cell to the negative charges on the inside of the cell. Sodium moves down its concentration gradient from high sodium outside to low sodium inside. And that concentration gradient is maintained by an ATPase. The sodium is maintained by a sodium-potassium ATPase. Potassium moves down its concentration from inside the cell to outside, and then the ATPase pumps it back in. The electrical gradient for potassium is tending to want to keep potassium in. The negative charges on the inside of the cell tend to attract the positive charge on the potassium. So I talked to you about water being able to tumble. It's got a negative charge on the oxygen, positive charges on the hydrogen. And so when you have hydrogen and another molecule over here with hydrogen. So oxygen tends to want to be attracted to the hydrogen, and the hydrogens are repelled, like charges repel. So this starts the molecule into spinning, and it's tumbling, set to tumble. And as it gets close to the negative charge on the phosphate head, then the positive charges attract, and as the oxygen gets close to gets in and it's close to the phosphate, that's going to repel, and that's going to continue it tumbling into the lipid part of the phospholipid. So the lipid is nonpolar. So as the water continues to tumble, eventually the hydrogens are going to get attracted to the opposite phosphate head, which is negatively charged. And that will pull it through, and then the oxygen will push it out into the intracellular fluid. So the hydrogens, they're not attracted or repelled by the lipid tails, but as the water is tumbling through, then the hydrogens will be attracted to the negative charge on the phosphate, and then gets far enough, gets close enough to the phosphate that the oxygen negative charge repels, and then now it's inside the cell. With water channels, those aquaporins that were discovered, these will allow a lot of water to move into the cell or out of the cell. Cells have aquaporins to allow water in maybe on one side and out on the other side. So if they're lining a duct, water usually comes in through aquaporins of one type and then out through another type. The aquaporin numbers can be upregulated. That means you can put more of them in the plasma membrane, or you can have them downregulated, meaning you pull them out of the plasma membrane. If they're upregulated, then more water moves. If they're downregulated, then less water is going to move. How do you up and down regulate? It's hormonally controlled. The movement of water, again, is tumbling. It has its own concentration gradient, and we have to always think about that first. So you come back to these examples. Why are they incorrect? Because they didn't think about water's diffusion. Water moves first. So you look at the solute concentration, high to low. Yeah, it's going to move from high to low, but it's going to move after water moves. So water, always you think about it as it's having its own concentration gradient. It's determined by the solute concentration. So if we have a solute concentration of 700 milliosmoles versus 300 milliosmoles, then water is high here and it's low here. Oops, sorry. So water moves from high to low. And if the solute can move, it will move after water moves. So here we've got 100% water. We got 90% water. Solute is taking up 10% of the space. So here water is in low concentration. Here it's in high concentration because, well, it's pure water, so obviously. This is not what's going to be happening in your body, except if you drink that water and bring it into your body, then you're going to lower your solute concentration even more. And if that solute concentration is critical to the function of your cells, you're going to be in trouble. So drinking pure water actually can kill you if you can't get rid of that water fast enough. So water is not something to fool around with. In the body, we've got a semi-permeable membrane. And if we put pure water outside a cell, water is going to move into that cell. That process is called osmosis. So osmosis is the movement of water molecules down its concentration gradient. So we talk about osmotic pressure. The osmotic pressure, the tendency for water to move down its concentration gradient into this situation down below here, from first scenario to second scenario, we've got water moving via osmosis into side 2, and the volume in side 2 is increasing. If this was a physiologic state and this was a cell, then the plasma membrane might actually burst if enough water moves across and goes into the cell. In this situation, all that's happening is the volume goes up on the side 2 and the volume goes down into inside 1 until the pressure on the surfaces are now equal. And then diffusion, net diffusion will stop. So the hydrostatic pressure is the pressure on the surface, which then is exerted into the fluid and across to the opposite side. So when the hydrostatic pressures, the water pressures are equal, then net diffusion will cease. So volume changes, absolute number of solute particles stays the same. So we got 1, 2, 3, 4, 5, 6 particles, hasn't changed. You had 6 particles in the first scenario, you got 6 particles in the second scenario. What has changed is solute concentration. That has changed. So delta means change. So that's a shorthand you need to become familiar with. Okay, so we're going to be talking about this tomorrow. So I'll come back to the next scenario, and we'll start reviewing all of this again tomorrow. So we're finished for today. Come back here, and we've got stop presenting. So you can leave.